Good afternoon. Welcome to my talk about image quality in publications. So the results that I present are from a collaborative meta-research study that a large group of us did recently, and I'm going to show you the group in a second. I'm Helena Jambor. You can reach me via Twitter or email, and I'm a trained biologist. I did a large imaging screen while I was a postdoc in Pavel Tomanczek lab at the Max Planck in Dresden, and today I actually focus on data visualization, and I teach and I'm an expert for showing for information design of biomedical data. And one aspect of that is, of course, also image data. So images have been used to communicate scientific data since antiquity. That's nothing new at all. And just to show you a really beautiful example, here is the page from a medical textbook from 200 years Anno Domini. And what you can see is different kind of scorpions, some of which are poisonous and some of which aren't. And what you can appreciate probably as a bioimaging expert is that the scorpions are shown in a very stereotypical manner. They all have the anterior towards the left and the posterior towards the right. And they are nicely arranged on the page. They have a good layout. We can get a very good quick overview of which guys we have to watch out for and which ones are maybe harmless. Obviously, in the last 2000 years, a lot of things have happened. Microscopies have been developed that allowed us to observe in much more detail biological specimen. Photography was developed, which allows the rapid documentation. No one has to draw or make copper plates anymore. And the very first photo that I observed in nature was from 1930, where a larva was shown with a photo. So that was the first photograph that I could find. And obviously, since then, the development of techniques have really sped up of what kind of images are shown in publications. We see movies that are possible with better cameras, with computers. We see three-dimensional data that is deposited, and of course this doesn't stop. So today the images are so advanced that we are able to observe the entire development of a fruit fly in a microscope in 3 dimensions, that we can see the heartbeat of a zebrafish heart, and we can see every single cell of blood that is passing through the heart. So the techniques have advanced massively. What also has increased massively is the number of publications. So around 800,000 new papers are published or indexed in PubMed every single year at the moment, which means every day we get 2,000 to 2,500 new publications that are appearing, and someone will have to read them. And around 23% is an estimate. 23% of these publications have figures with some kind of images. Obviously, these images are not all movies. There are different kinds. We have drawings, we have computer animations, we have Western blots, we have photographs, we have microscopy data. And with this massive amount of publications and huge numbers of images that are flooding really the biomedical community, obviously we also have seen in the last 20 years or so really an increase of the problems that arise with images. So we see misleading images that are being published. We see a lot of non-expert users of microscopy technique that are beginning to use it. We see a huge technology diversification that makes it problematic maybe for some PhD students to rapidly learn techniques. So image manipulations is actually in the news since I'm aware of it since around 15 years. What we are mostly speaking here about is the intentional image manipulation. That usually gets a lot of attention. There are many prominent cases, and some of which are really ending careers. We've seen platforms such as PubPeer appearing with their milestone publications. Everyone probably knows the blog by Elizabeth Big that is really tracking down systematically cases of intentional image manipulation, intentional image manipulations. And some of the famous examples are shown here, where images were spliced together, where image duplications appear, where parts of photographs are duplicated and reused as control, etc., etc. And Elizabeth already in 2016 quantified this a little bit and showed that really with the number of publications rising, we simply get an automatic rise as well in the number of manipulated images. There's, however, a second aspect of image manipulation. So not all of them are intentional, some are simply accidental. And they appear with the erroneous handling of maybe a microscope or a post-imaging erroneous handling of the image data files. They get a lot less attention since it's not intentional. And they are still very problematic actually for the scientific legacy. If we think that there are many of those in publications, we really need to worry about whether the data is at all useful in the future. And both image manipulation, intentional and accidental, has been addressed by the Council of Scientific Editors. So they've published a white paper on promoting image integrity, actually promoting integrity in scientific journal publications. It was about a lot of things, and one aspect of that was also image manipulations. So they've defined what to do when they see such cases. But this is not all. In fact, I'm teaching since 6 years data visualization or visual communication to biomedical graduate students mainly. And I see a lot of insecurity, and I see a lot of very, very basic questions that are being asked. Some are here. Some isn't cropping an image actually misleading already? Can I still measure the intensity after I turn an image upside down or by 90 degrees? I did not know that I could change colors in image. I heard that turning changes pixel intensity and is therefore falsifying results. Am I allowed to show GFP in red, etc.? So what I observe in these courses is that students are really well aware that splicing together images and pasting in Western blot bands is absolutely not okay. So they know about intentional misleading image, and they usually obviously don't do it. But there's a really great insecurity about what is allowed and what is not allowed. And I think we need to teach basics to the students, and we need to think of how we can integrate them better into a scientific discourse. In fact, around the time that the students started asking such questions, I was wondering how good are images presented in publications if these are the questions that I get in courses. And around that time I met Tracy Weisgerber. So we were both eLife ambassadors. Tracy has done a couple of prominent meta-research studies already. I have had a lot of experience teaching information design and data visualization, and our joint idea was really to quantify image publication standards in a meta-research study. And the group that formed was from the eLife Ambassador program, so we gathered a lot of people that were really interested in doing collaborative work for the good of the scientific community. And I just want to show the group already. So the team consisted of 19 scientists from 9 different nations, and here are all the names and all the institutes that are part of, and some have allowed me to show their photos. So this was a big team effort. So we wanted to find out what is the status quo of image presentation in papers. Is it really bad, or just get some numbers? Obviously, we couldn't screen 800,000 publications. We had to somehow make a compromise on what we wanted to screen. So we decided on the following sample set of articles. So we chose 3 representative biomedical fields, fields in life science actually. We chose cell biology since it's very heavy on microscopy and imaging. We chose plant science, and we chose physiology because it's a very prominent field where a lot of medical people are also publishing. For these 3 fields, we chose to screen the top 15 journals. We just decided on the top 15 based on the impact factor. The top 15 journals that are publishing original research. So we obviously wanted to only screen original research articles. And we decided on a pretty much arbitrary time frame. We wanted to look at all publications that appeared in April 2018 in any of those journals. And the number that we then settled on is close to 600 papers. We excluded images, we excluded papers that had images that only had Western blots or computer-generated images, since these have entirely different kind of requirements for visualization in publication. And just to give you an impression, these are the journals that we screened. For cell biology, this obviously includes Cell, Nature Medicine, Cancer Cells, Cancer Stem Cell, Nature Cell Biology, etc. So the prominent journals in cell biology everyone knows. So these are not obscure journals, but these are really representative journals for the field of cell biology. And I cannot judge for plant science and physiology if that's the case too, but here are the names and you can inspect this further. So next, now we had a sample set of articles that we wanted to have a look at, but we wanted to do it systematically. So we didn't want to just eyeball our own impression, but we wanted to really get good numbers. So we decided to develop an abstraction protocol. The abstraction protocol basically is a set of questions with defined answers, where each abstractor, so each expert that is looking at the publication, goes through and can systematically decide, for example, image type, what kind of image type it is, whether the images have scale bars, insets, etc., etc. So the abstraction protocol is obviously published along with the paper as well, and basically every paper was screened by at least 2 abstractors, and if they couldn't reach a consensus, more people were asked to look at the papers as well. So at the end of the day, every paper was seen by at least 2 people. So what kind of results? What did we find? First of all, the image types that we focused on, I already mentioned this before, were microscopy images and photos, but also EM pictures or clinical images. And we looked at the categories physiology, cell biology, and plant science. And just to introduce here already the color code that is used throughout a talk for these 3 sections. So not surprising, since we already wanted to focus on they all had images. That was the criteria for inclusion, and most of the images, 90% in cell biology and physiology, were indeed microphotographs, so confocal, etc., images from some kind of microscopy instrument. And in plant science, this was very heavy on photos as well. The 3 other image types that we screened, clinical image or electron microscopes, appeared but were not prominent numbers. So to summarize that, most papers with images had microphotographs or plant Plant Science had photos. So the first thing that we then screened for was scale bars. So I think most of us in the bioimaging community are very well aware that scale bars are the only way to relate an image to reality, and they should be included each time we publish an image. However, when we look at publications, we see many times cases such as this. For example, that no scale bar is actually shown in an image, that the scale bar is shown in a color that is indiscernible from the background almost, so you can see it in blue here. Very often scale bars are entirely illegible because the images were compressed too much, or the scale bars are so thin or small that they're not visible at first glance. So if you zoom in here, you can see at the lower right hand corner actually a very small scale bar. Sometimes scale bars blend into the background, for example in EM microscopes or in histograms. So how common is this? So these are we scored either that images had complete scale information, partial scale information, meaning some images had a scale bar and others didn't, or all images in the paper were entirely absent of scale bars. So within these groups we made further subcategories, and you could read the details in the paper. But basically the take-home message is that only 30 to 40% of the papers with images had complete scale information. The majority had either partial scale information or, in fact, no scale information whatsoever. So to summarize, around 45 to 70% of the papers, the images have either no or only partial scale information. And that was a big surprise to me, actually, because I roughly thought that maybe in 10% of the cases the scale bar is missing. But in fact, this is much more prevalent and slightly different in different fields. But a lot of images do not have enough scale information in order to be understandable to all audiences. So scale bar obviously is the first key factor in order to relate images to reality, to give the audiences a clue of what is shown. But very, very importantly, most microscopic images have some kind of colors. So we were wondering if colors are shown, for example, either we have a 2-color image or images with 3 colors. We were wondering how many people are paying attention to making sure that these colors are still discernible to a colorblind person. So if we render, for example, these 2 images, you can appreciate that red-green obviously cannot be distinguished by the atypical version of color blindness, and 3-color images similarly cause problems. So in screening, we always checked for colorblind vision with the Color Oracle, which is a desktop application that can render the entire screen in different kind of colorblind visions, and that allows you to judge quickly whether images are visible to all audiences. So what are the data here? We asked if image features themselves are visible to colorblind. In other words, if a cell type or a staining is visible, and the data you can see here. So that in many papers actually had problems with that only some of the images are visible to colorblind people. So most papers, more than 50% of the papers still were okay in every aspect, but a substantial amount had problematic images that would not be visible to colorblind audiences. And we also looked at whether the labels are visible, because indicating the color is usually done with a colored label. that, for example, blue is DAPI and red is some kind of actin stain and green some kind of GFP or something like this. So if you have colored labels, they should obviously also be visible to colorblind people. So in summary, of our close to 600 papers, 350 papers had images with adjustable color, meaning they were confocal microscopes or something like this, image files where the colors were separated in different channels and could have been adjusted in post-processing. And of those, only 50% were of the papers had images that were not or only partially accessible to colorblind people. Again, quite a surprising number, given that this gets a lot of attention in the bioimaging community already before us. The next aspect that we wanted to screen had nothing to do with scale bars or color blindness, but many times images also get annotations and they have to be legible to all audiences. So for example, very often we see arrows, we see dashed lines, we see letter codes, or we see any kind of lines that point to regions of interest. But then of course we also have often colored annotations so that certain areas are filled in order to be visible as a nucleus or a sub-region in the nucleus or something like this. So there are lots and lots of different kind of annotations in images. And I will not go into the details now of every aspect here, but just to summarize, the 3 questions that we screened were whether the colors are clearly described in the figure or the legend. So is the annotation of the color complete? We also asked if all the species and tissues and objects are actually described in the legend. So if you open the figure, can you actually figure out what kind of object you're actually seeing at all? Or do you really have to read the methods section for that? And the 3rd question about annotations was whether all the labels, such as arrows or asterisks or symbols, are really clearly described in the figure legends. And again, what we could see here was that there is a lot of room for improvement of systematically explaining all annotations. So in many cases, not all image symbols used were explained in text, which really make it hard for audiences to read papers. One last aspect that I want to quickly comment on is that of insets. So we very, very often see obviously insets in papers. And sometimes they go wrong. For instance, what we have observed is that sometimes insets appear from a random region of the image, so they were wrongly placed simply, mostly I assume by accident. Sometimes we see images where it's absolutely not even tried to indicate where an inset is coming from. Sometimes the inset obstructs the actual data, so we have sometimes insets that are sitting on top of the data and we cannot really understand anymore whether important data is maybe concealed by that. And what we asked here is if the insets are accurately marked in the original image and if they are explained in the image or the legend. And again, we made a lot of distinctions that are maybe not quite relevant right now. But what we observed was that a substantial number, again, of the images, of the insets that are shown in images are partially or inaccurately marked, meaning it is unclear where the inset actually comes from. And they're also not sufficiently explained in the legend. So 30 to 45% of the insets are not or only incompletely marked in the original image, and only 50% are explained somewhere in the legend. So to summarize, 50 to 70 papers of the papers that we screened had some kind of images. These were around 600 that we then looked into more detail. And of those, 45 to up to 70% had not enough scale information, not sufficiently explained all scales. 30 to 50% had colors that were not adjusted to be accessible also by colorblind readers. And 30 to 50% lacked explanations of, for example, inserts, the colors, the objects, the arrows, etc., etc. And if we take that together, I asked one of the co-authors the question recently, how many of the papers were good in all of our screening questions? So how many papers passed in every, were doing fine on color and fine on scale information and had enough explanation? And the realization was that only 10 to 20% of the papers across all different, across cell biology, physiology and plant science, only 10 to 20% of the papers had fully legible images. And I want to point out that we were not very strict. So our standards are really the bare minimum of what needs to be done to have a legible image. They were not even addressing methods, and they were not even addressing really details. And we recently learned from a paper published in eLife by Marquez, Pengo and Sanders, that who had in parallel to us started screening, but they didn't look at image legibility, but instead they looked at imaging methods reporting. And they also found that a huge number of papers insufficiently explained how microscopic images were acquired. Again, probably diminishing the numbers of papers that are fully legible, well below 10%. So basically our take home is that we think it needs to be we need to substantially improve the quality of images in papers. Why? Not just to make Helena happy or the bioimaging community, but we think that better legibility allows all audiences to really gain insights from papers. So our papers become more valuable if we have understandable images. Again, think about the numbers. Around 2,000 papers are published per day, 500 of which contain images, 20 of which have probably image manipulations based on the data by Elizabeth Dick, and 400 of which are not fully accessible or not fully leverageable based on our data. So we believe that improving the legibility of images doesn't only make it more accessible to your readers today, but in fact they will become a better legacy of the scientific community. So we have to always think that maybe, like we are reading medical textbooks from 2,000 years ago, maybe someone is in fact still interested in the scientific data that we are acquiring today in the future. And if images are not legible now, they will not miraculously become legible in future, meaning the scientific data that is published is lost for the greater good of humankind. And obviously also we would like to see a better quality for the advent of image archives. So if we start to systematically collect images and image archives, and with the intention of one day maybe automatically screening data in these image archives, we really need to make sure that the quality is substantially improved and that we have a quality that is actually machine readable. If we have too many images that are not legible, no AI tool will be able to gain substantial insights at all. So we think there are very many good arguments for improving image quality, And we didn't want to stop there, but obviously we are thinking of how to do that. What can be done to improve the images that are published today? So obviously we can always start with journal guidelines, and we can lobby among scientific publishers that they expand the checklists for copy editors. So at the moment they rarely contain questions about image legibility. We can also bully, or no, not bully, we can also argue a case that journals have improved checklist for reviewers, but we should not forget that reviewers already have to do a lot of work and might not be willing to do even more. Journals could set up automatic screenings, for example, things like color legibility or accessibility to colorblind can systematically simply be screened with tools such as Color Oracle. And some ideas are also revolving around whether we really need figure limits, page limits, and page charges, or whether these are not obstructing or whether these are not hindering people to make really nicely legible figures that are large enough for people to see. And obviously journals have already started, but should continue to encourage the use of public image repositories, because here the scientific community can set quality standards. Before images are deposited in an image archive, they have to fulfill a certain number of criteria. What else can be done? Grant agencies can have a small role, probably. They can require legibility of figures and images in grants at least. They can fund training, and there are probably more ideas that we can discuss also in the QuiRep meetings maybe. But we believe actually that the biggest hope is that the scientific community actually gets engaged, because this is not a problem that can be solved by journals or grant agencies. So the scientific community needs to become a little bit active here. The basics, the very, very basics of producing legible images are really quick to learn, and I have experienced from 6 years they are really rapid to teach. And the good thing is they have a really lasting effect. So responses that I sometimes get from students and also some people in the eLife meta research team is that I cannot stop anymore to check colors. So once people know red, green is problematic for colorblind, they cannot stop seeing it. And they also commented that doing this project really changed how they can see images forever after. So once something is seen, it cannot be unseen. So the scientific community as reviewers obviously can screen images and figures for their legibility, and they can comment as reviewers requesting that images are improved. As innovators, scientists can support the imaging archives and help to develop quality standards of images that go into these archives. And as teachers, obviously, they can train students. And obviously there is a lot of training material for different aspects, but we felt what was lacking really was basic teaching material for explaining what constitutes a legible image. So because of that, the half of our paper is actually devoted on on giving examples for how cropping, how colors, and how annotations can be done in a legible manner. And here's again the link to the paper on the bioRxiv, where we have a lot of resources and a lot of images that can be used for teaching how to make legible images for publications. We also, I was also involved in developing a training opportunity for basic image processing. So together with Christopher Schmid, who's shown here, he's an image analysis specialist in Berlin. And like me, he's doing a lot of teaching, and he sees a lot of the really basic mistakes that novice users make, or a lot of basic questions that novice users still have. So together, Christopher and I developed a really basic, simple workflow for image processing that is aimed at novice users. So we really just point out the different steps from what you can do after you get the microscope TIFF file in order the different steps that you need to take and you need to decide on in order to have a publishable image at the end that is legible. So we describe the workflow, and we describe how to do it in Fiji, so in open source image software that everyone here knows. And accompanying this workflow, we also published a workflow for image publishing. And the main aim of the paper was really to publish 2 cheat sheets that we think can circulate and can be used by students all over, or actually not only students. Everyone who's new to microscopy needs to start thinking of how to make a legible image in the end. And we believe that these cheat sheets, which will be available very soon in F1000 research, are one way to get there. So to summarize this up, if we think about image data, we need to really think about the data visualization cycle. So on the one hand, we have the encoders, the people at the microscope that obviously need to think of the object and how to collect data and how to operate a microscope. But what we would like to kind of bring home is that also this is a cycle. It doesn't stop there, but once you've acquired the data at the microscope, you need to think of how to visualize it and how to visualize it such that an audience can decode the visual code, that can understand the annotation, that they can understand the colors used, and only then they are able to actually interpret the image data in order to gain insight. So we hope that the focus switches from only thinking about the encoder side and actually training also everyone to think about the audience who will in the end read a paper and look at an image and wonder what is shown in the image at all. And with that, my talk ends. If you have questions, I hope that I will now be able to join via phone and answer a couple of your questions. If that will be technically impossible, please feel free to reach out on Twitter or by email. And I think some of you I will meet shortly in the QUAREP group for image quality in publications. Thank you very much.